Our next lecture is from Dr. Lewis Cantley, the 2015 Canada Gairdner International Laureate, and the Margaret and Herman Sokol Professor and Director of the Sandra and Edward Meyer Cancer Center at Weill Cornell Medical College and New York Presbyterian Hospital. Dr. Cantley received this award for his discovery of phosphoinositide 3 kinase, a critical component of the cell signaling machinery that plays a key role in the normal functions of proliferations and growth and is deregulated in diseases such as cancer and diabetes. His discovery showcased a new language of how cells control themselves internally. He has received many prestigious awards, including the 2013 Breakthrough in Life Sciences Award, and was elected to the Institute of Medicine in the United States in 2014. Please join me in welcoming Dr. Cantley to the stage for his lecture entitled PI3 Kinase and Human Disease.
It's a pleasure to be here today and see many old friends from Toronto, and of course a great honor to receive this award. So I will be talking about PI3 kinase, its pathway and its role in disease. And the talk really will have two parts to it. In the first part, I'll talk about an overall view of PI3 kinase and what its role in various diseases are, and something about the biochemistry of this enzyme. And then in the second part, I'll talk about some rather recent work of trying to translate this basic discovery into cures for patients with cancers. So PI3 kinase we discovered in the mid-1980s as an enzymatic activity that co-purified with a variety of oncoproteins. And the reason we were looking for this is we, at that time, were working on membrane transport systems, proteins that would take molecules across cell membranes, particularly ions. And so we had discovered that as we reconstituted some of these proteins, they were very sensitive to the lipid environment, and that phosphorylated forms of phosphatidylinositol had differential regulatory abilities on those proteins. And so no one had purified an enzyme that would phosphorylate phosphatidylinositol at that time, although two phosphorylated species were known to exist, one in which the phosphate was at the 4 position, and one in which it was at both the 4 and the 5 position. So these were called PI4P and PI45P2. And ironically, those two lipids were actually discovered the year I was born, 1949. And the order in which they were put on the cell was described in the 60s. So we assumed that the activity that we were seeing and the regulatory effects that we were seeing were due to one of these two species, and we sought to understand what those enzymes were and looked to see what what was regulating them. And in the process, discovered that a PI kinase was co-purifying with various oncoproteins that cause cancers in mice and chickens, including the VSARC tyrosine kinase. And as we purified the enzyme and began to characterize it, we realized it was quite different from the major PI4 kinase we could also detect in cells, although no one had purified any of these to homogeneity at that time. But as we looked at the product, we realized it moved slightly different in a thin layer chromatography system, and realized it was chemically different from PI4P, and ultimately went on to show it was phosphorylating the 3 position of the inositol ring rather than the 4 position. We then went on to show that this enzyme that was co-purifying with the oncoproteins could actually put phosphate on the 3 position of the bare PI molecule, but it could also put at the 3 position of the previously phosphorylated 4 molecule, 4P molecule, or the 4,5P molecule. And the lipid I'll primarily focus on today is PI345P3. The monophosphorylated PI3P is produced by the enzyme that we heard about in the previous lecture, the so-called VPS34, turned out to be in yeast responsible for putting the phosphate to 3 position. But I'll focus on this triply phosphorylated lipid that was being generated by various oncoproteins. Now, about 10 years after we discovered this enzymatic activity, a phosphatase was identified by positional cloning on human chromosome 10 that was called P10. It was predicted to be a phosphatase based on the primary sequence and thought to be a phosphotyrosine phosphatase It had and does have low phosphotyrosine phosphatase activity. But Jack Dixon, who's really a brilliant enzymologist, who had worked on protein phosphatases for much of his career, realized that the turnover number for this enzyme was far too low to be a protein phosphatase, and sought other substrates and discovered that it could dephosphorylate the very same position on the inositol ring that PI3 kinase phosphorylated. And that then, since this had been identified as a tumor suppressor gene by positional cloning, implicated that in human cancers, not only could this lipid, this triply phosphorylated lipid, be made due to activation of PI3 kinase, but also by inhibition or loss of function of the phosphatase. Now, PI3 kinase obviously did not evolve for the purpose of causing cancer, but we can understand quite a bit about what its role is by looking at where you first see this triply phosphorylated lipid produced in nature. So Saccharomyces cerevisiae, as I said, make the PI3P, and that's essential for the autophagy story that you just heard, but they don't make this PIP3, the triply phosphorylated lipid. You only see that in multicellular organisms. And we now know that what it's doing in those multicellular organisms is telling cells when to grow in the organism. So with a multicellular organism, as the organism eats, there has to be a decision of which cell in that body takes up nutrient and grows at what time during development. And what PI3 kinase does is allow that to be coordinated so that food that's taken in only goes to the right tissue at the right time to properly develop each organism, each organ. And so its primary function is to respond to insulin, or IGF-1, which is highly conserved back to flies and worms, and the insulin receptor, highly conserved. And that's essentially the only way to activate it in those multicellular organisms. Now, in humans, we can activate PI3 kinase by a variety of growth factors, but the evolutionary precursor to this whole pathway is downstream of insulin, mediating glucose uptake and metabolism. And as long as P10 is function in those organisms, every tissue only grows when it's supposed to grow, and you have homeostasis. Now, what happens, we now know in human disease, is a failure of insulin to be able to activate PI3 kinase results in type 2 diabetes. Insulin resistance initially, ultimately progressing on to type 2 diabetes as the pancreas becomes incapable of making sufficient levels of insulin to overcome this insulin resistance. And so we still, today, don't fully understand all the mechanisms by which type 2 diabetes can arise, but it's clearly a consequence of overeating, overstimulating the pathway, and the attempt to turn this pathway back off that develops this disease. And this can, of course, correlates with metabolic syndrome. This is the most rapidly growing disease in the Western world today, in fact, in all the world, as people are tending to overeat. Now, in contrast, if a cell acquires the ability to activate PI3 kinase inappropriately, and this can happen, as I'll show you, by mutations in the gene encoding PI3 kinase itself, it can be due to loss of P10, it can be hyperactivation of the growth factor upstream, or even just high ambient levels of insulin can turn this pathway on dramatically if your tumor cell happens to have insulin receptor. And so we know that all of these things can happen. They'll drive very high levels of PIP3. In fact, most cancer cells have this lipid very highly expressed, very high levels of this lipid. That turns on glucose uptake and anabolic processes that drive cell growth and allow cells to survive in a variety of environments and ultimately can progress into cancer. Now, it turns out that PI3 kinase, in fact, the gene that makes this triply phosphorylated lipid in most cells, called PIK3CA, can also be mutated during early embryonic development. And there's a disease called localized hypertrophy. It manifests in a variety of different ways. It could be just a single toe that takes off and grows continuously throughout your life at much higher rates than surrounding normal tissue. It can be an entire limb and a leg, as you can see here. It could be a part of your neurons in your brain, resulting in an enlargement of the brain, outgrowing the skull, in fact. And as these were ultimately realized to be a consequence of a mosaicism in which sometime during development, one cell in the early embryo picked up a mutation. And so as these tissues were being sequenced, it turns out in the vast majority of cases, it's an activating mutation in PI3 kinase that's causing this disease. And in fact, the mutations that are occurring are exactly the same mutations that we see in cancers where PI3 kinase is mutated. Now, these aren't true cancers, but if an additional event should happen that allows these hypergrowing cells to maintain stem-like character, they will progress into cancers. So how does production of this triply phosphorylated lipid turn on cellular growth? And we understand that in great detail. I don't have time to get into all of that today. But for example, when insulin binds the insulin receptor is a tyrosine kinase. It autophosphorylates itself. Those which is what these little P's with circles around indicate. These are phosphotyrosine sites on the receptor. And this recruits an adapter protein called IRS1, or the family of IRS1 adapter proteins. They themselves get phosphorylated at multiple tyrosine sites. I only showed 2 of them here. But in fact, there are 9 sites on this protein that are in a specific sequence context of tyrosine, methionine, X-methionine that creates an optimal binding site for the regulatory subunit of PI3 kinase, P85. The PIK3CA gene encodes the catalytic subunit, which we call P110 for its molecular weight. So this complex is recruited to the insulin receptor through this IRS1 adapter protein, which of course resides at the plasma membrane, bringing the enzyme to the plasma membrane, activating it, and allowing it to convert PI45P2 to this triply phosphorylated lipid locally where insulin receptor is being activated. A host of proteins in the cell have domains in them, plex homology domains, that will directly bind to this lipid product, and that allows them to come to the plasma membrane. And 2 of the proteins that we know quite a bit about include 2-serine-threonine kinase, the protein called serine-threonine kinase called AKT, and its upstream activator called PDK1. They both directly bind to the products of PI3 kinase. This brings them together and allows PDK1 to phosphorylate AKT and turn up its serine-threonine kinase activity. It then phosphorylates a host of proteins in the cell. In fact, once phosphorylated, it actually gets phosphorylated at 2 sites, not only by PDK1, but also by TORC2, which you'll hear about from the next speaker. And that turns on the activity, so AKT doesn't have to stay at the plasma membrane at that point. It can wander off and phosphorylate numerous other locations in the cell. But of the more than 150 proteins known to be phosphorylated by AKT, several of them are highly conserved back to flies and worms and can begin to explain everything that insulin does in the cell, including increasing glycogen synthesis, turning off gluconeogenesis in the liver, turning on protein synthesis, and stimulating glucose uptake into muscle and fat. So I'll talk a bit more about this protein synthesis regulatory mechanism because it will tie into to Michael Hall's talk after me. And I'll give you a little introduction to his talk. As long as P10's around and can turn off PIP3, everything shuts back off and the cell comes back to homeostasis after this brief period of responding to insulin. Now, it turns out that there are some rare diseases where mutations in the regulatory subunit of PI3 kinase can prevent PI3 kinase from getting recruited to the insulin receptor. The insulin receptor still binds insulin, gets activated, phosphorylates itself on tyrosine, phosphorylates IRS1, but it can't recruit the PI3 kinase. And the consequence is you have severe insulin resistance to the stage that looks very much like type 1 diabetes in that insulin doesn't work at all. And as a consequence, this disease, which is called short syndrome, results in remarkably small children who would be misdiagnosed as type 1 diabetics. Now, a bit about protein synthesis links. So when PI3 kinase gets activated and activates AKT, this will result, among many of the substrates of AKT, phosphorylation of a protein called TSC2, which we showed several years ago is a negative regulator of mTOR signaling. And it does so by regulating a protein, a RAS-like GTP binding protein that directly binds to mTOR and brings it to the lysosome where its major function is in the context of what's called the TORC1 complex. And you'll hear more about the difference between TORC1 and TORC2 from Michael Hall. So TOR complex is conserved, both of these TOR complexes are conserved all the way back to yeast. And this so-called TORC1 complex controls protein synthesis, and we understand in some detail how that works. But it also independently regulates lipid synthesis and controls a number of transcription factors, including HIF and MYC, by a rather indirect mechanism that affects glucose uptake and protein synthesis in the cell. Now, one problem with the complexity of this is that although normally this pathway is typically regulated downstream of insulin and other tyrosine kinases, but mainly insulin IGF-1 signaling, it can also be activated by an independent pathway involving the so-called MAP kinase signaling network, which converges again on this TSC protein. TSC2 gets its name from the fact that it is a gene that's defective in a disease called tuberous sclerosis, so loss of function of TSC2 allows mTOR to be constitutively active, and inhibitors of mTOR can then be used to treat that disease. And they're also beneficial in cancers because they can slow down the growth response due to activation of PI3 kinase. There are other sensing mechanisms of mTOR that I don't have time to get into that sense the energy state of the cell and the availability of amino acids. And as I say, the bottom part of this pathway is conserved back to flies and worms, and the regulation by tyrosine kinase is unique to multicellular organisms. So it turns out that also everything in this slide that is, everything that is blue is a tumor suppressor, everything is red is an oncogene. So gain of functions of the things in red will turn on cancer growth, and loss of function of things in blue, which are typically the brakes on the system, will result in cancers as well. Now, if we ask how often is PI3 kinase, PIK3CA, the gene that regulates this form of PI3 kinase regulated by insulin, how often is it mutated? It is, in fact, the most mutated gene, oncogene, in all of cancer, even more frequent than RAS, which is also extremely frequently mutated gene and in the same pathway. In women's cancers in particular, you see very high rates of mutation of this gene. Uterine cancers, endometrial cancers, the majority of them have an activating mutation in PI3 kinase indicated by green. The reds indicate amplification of the gene. You see this very often in ovarian cancers. Cervical cancers have both activating mutations and amplifications, and breast cancers primarily have activating mutations. If we ask how often is this entire network or some component of this network mutated in cancer, then we see that all the cancers that are responsible for the majority of deaths in the Western world are dominated by mutations in this signaling network that I showed you, including prostate cancers, lung cancers, colorectal cancers, and glioblastomas. And in fact, glioblastomas and endometrial cancers, virtually every single tumor has at least one, and in many cases, multiple genes. That's what the gray means, is that many components of this pathway in the same cell are being mutated in that disease. So as a consequence, pharmaceutical companies have not ignored this observation and have begun to develop inhibitors of everything in this pathway that can be inhibited. I left off some well-known cancer drugs that hit tyrosine kinases. There's a lot of those now, EGF receptor inhibitors, ALK inhibitors, HER2 inhibitors. And recently, inhibitors of RAF and MEK in the MAP kinase pathway have been approved for treating melanomas. And inhibitors of RISC and ERK are also in clinical trials. But there are also numerous inhibitors of PI3 kinase in clinical trials. More than 20 PI3 kinase inhibitors from multiple companies have gone into clinical trials. The only drug thus far approved is a drug called Adeleza, and I'll tell you a little bit about that drug in a moment. This drug is made by Gilead. But several of these other PI3 kinase inhibitors are now in approval trials for breast cancers and other solid tumors. AKT inhibitors are also in clinical trials, and inhibitors of mTOR have already been approved for treating cancer, everolimus, for example, from Novartis. Activators of AMP kinase are also being explored. There are more than 200 cancer trials in which metformin, which can activate AMP kinase, is being tested for its ability to suppress tumor growth. Now, if we ask in humans, how many genes are there that can make this triply phosphorylated lipid, PIP3, there are 4. There are actually 8 PI kinases altogether in humans, including the VPS34 homolog that you heard about, but 3 other genes that will make PI3P and 34P2. But the only ones that will make PIP3 are these 4 genes. PIK3CA is the only one that's really significantly mutated in cancers, although occasional mutation PIK3CB is seen. And it turns out that PIK3CB turns out to be critical, for example, in prostate cancer if you lose PTEN. So even though it's not mutated, it's still a good target for treating prostate cancer, and that's being explored. PIK3CD is the only PI3 kinase whose drug has been, in which a drug has been approved. This Adeleza that I already mentioned is an inhibitor of the protein product of PIK3CD. PIK3CD is found primarily in hematopoietic cells, T cells, and B cells. And interestingly, there are point mutations in PIK3CD that are gain-of-function mutations, analysis to PIK3CA. These are actually found in germline. They're transmitted in germline, but they don't result in cancers. They actually result in immune deficiencies, in that they exhaust the T cells. The T cells grow so fast that they become exhausted, and except for the Tregs, which you'll hear about later, can still stay around, but the other T cell compartments are eventually dissipated. It turns out that B cells, however, are very dependent on the PIK3CD, and that's why this drug is useful in B-cell lymphomas, and I'll show you a little bit about that in a moment. Obviously, inhibiting the gene that's product that's mutated is important in numerous alpha-specific inhibitors, including BYL719 from Novartis are in clinical trials. There are pan-PI3 kinase inhibitors, BKM120, and I'll mention a bit about both of these drugs in clinical trials that we're running currently. And then there are inhibitors that hit alpha, delta, gamma without beta, and numerous other approaches. In fact, every one of these genes is being, drugs are being developed against all of these diseases for proteins for various diseases. A bit about the PI3 kinase delta. The very first knockout we did of PI3 kinase we did back in the 1990s, and we did a hematopoietic cell specific knockout of all the class 1A PI3 kinases by eliminating the regulatory subunit and therefore shutting off all of those enzymes simultaneously. And to our surprise, we didn't see much of an effect on T cells. In fact, only T cells that were affected were Tregs that were suppressed. But in the B cells, we could not get B cells to grow at all. So triggering the B cell normally stimulates B cell growth. This is mediated not only by AKT, but a tyrosine kinase that also binds to the product of PI3 kinase called BTK. And point mutations in BTK that fail to bind PIP3 result in an agammaglobulinemia due to a defect in being able to trigger PI3 kinase. So these discoveries led to the rationale that an inhibitor of the PI3 kinase delta or BTK might be useful for treating B-cell lymphomas, not because those genes are mutated, but because they're required for that lineage of cell growth. And so Calistoga, a company in California, developed a delta-specific inhibitor and tested it in B-cell lymphomas, and sure enough, it was very effective in B-cell lymphomas. Consistent with the knockouts, there was no effect on T-cell leukemias, but the B-cell lymphomas responded dramatically, particularly chronic lymphocytic leukemias, which are a B-cell derived leukemia, where the waterfall plot, which is telling you the total tumor burden in the patient, is showing a dramatic response in almost every single patient. This is the type of phase 1 study that you dream about in cancer. Virtually no drug ever looks this remarkable in phase 1. Indolent non-Hodgkin's lymphoma was not quite as responsive, but you can see there's certain responses there, and this drug is still continuing to be studied in this disease. And in mantle cell lymphoma, also a B-cell derived lymphoma. There are trials going on at Weill Cornell that my lab is participating in preclinical studies to support in drug combinations. But this is an example of a patient with chronic lymphocytic leukemia who was being treated at Weill Cornell. She was only 38 years old. She'd already run out of options at that age. She had been treated with standard of care and was progressing, and the swelling of the lymph nodes here is the indication of her disease. This was developing quite rapidly, and she really didn't have long to last, and went on a phase 1 trial of Adeleza. And this is what she looked like only 3 months later after going on this drug. So it was really quite remarkable for her. But unfortunately, as single agents or even in combination with rituximab, these are still not cures. They give the patient several years of very healthy life, but ultimately the disease comes back, and we're still beginning to understand the mechanisms of resistance, and also beginning to now take this drug, since it's approved, into early stage treatment rather than wait until they failed all other therapies, where we're hoping to have better effects. So there's a lot of activity of looking for other novel drug combinations at Weill Cornell to try to improve the outcome for these patients. As I say, this drug was approved a little over a year ago. So I now want to spend the last few minutes talking about solid tumors, breast cancer in particular, and how a team of scientists from multiple institutions that I put together a few years ago and funded by Stand Up to Cancer and the AACR, American Association of Cancer Research, has been attempting to figure out how to use PI3 kinase inhibitors for treating breast cancer. We received $16.5 million to put together this team. Gordon Mills was my co-leader, and Charles Sawyer's originally also on the team. Charles is in gray here because he abandoned us after 2 years to start a prostate cancer Stand Up to Cancer team. But he was really quite instrumental in helping us initially put the team together and design some of the clinical trials we did. These are the other site leaders, Dana Farber, Tom Robertson, Eric Weiner, Ramon Parsons, who discovered P10 by positional cloning, was on our team from Columbia and now at Mount Sinai. David Solit took over Charles Sawyer's leadership at Sloan Kettering and Carlos Artiega at Vanderbilt. And Jose Baselga, at the time we started the team, was our leader at Vall d'Hebron in Barcelona. Jose moved to Mass General and brought Mass General into the fold. And then he and I both at the same time moved to New York, where he went to Sloan Kettering to become physician-in-chief there. And we also started a journal together. And so we've had a long-term collaboration with Jose. So if we look at breast cancer and ask what's going on with this network of signaling that I showed you earlier, you see there's a lot of activity going on. You probably can't read the numbers in this chart, but you'll notice there's a lot of bold numbers. The bold indicates that at least 20% of the tumors of that category have a genetic aberration in that gene. And so you can see a lot of bold here in P53, a gene that's the most frequently mutated tumor suppressor gene, but you also see loss of P10, particularly in triple-negative breast cancer. INPP4B is another tumor suppressor, the phosphatase that also degrades PIP3, actually degrades PI3-4P2 that we discovered a few years ago, very frequently deleted in triple-negative breast cancer and ovarian cancer. PIK3CA is mutated very frequently in ER-positive and HER2-positive breast cancer, but relatively rarely, only 8% in triple-negative breast cancer. So in the end, we ended up doing clinical trials in all these settings, but I'm only going to tell you about one clinical trial that we have a lot of excitement about, and that's in triple-negative breast cancer. And what we noticed as we put this chart together is that triple-negative breast cancer actually more resembles ovarian cancer with regard to genetic aberrations than it does the other two subsets of breast cancer. And in fact, these two cancers have in common that they are the two most common events that happen in patients who have germline BRCA aberrations. They get ovarian cancers or triple-negative breast cancers are what one most commonly sees. And that's almost certainly related to this common genetic set of aberrations that occurs in these two diseases, although we don't fully understand this yet. So in order to try to understand how we might treat this disease and whether this disease, because of the common loss of P10 or an occasion activation of PIK3CA and the loss of INPP4B, might be responsive to PI3 kinase inhibitors, we developed a mouse model in which the BRCA gene was deleted, specifically in the breast, and P53 was also deleted in the breast. So these are events that you almost invariably see in BRCA germline patients, loss of these two genes. And then we ask, first of all, do you get a cancer? And the answer is yes. Actually, this model was not developed by us, but by Yonkers Laboratory. It already demonstrated that these mice get tumors within about 7 months. And we began to characterize these tumors, and although it takes 7 months for a tumor to appear, from the time you can palpitate a tumor in one of the breasts of the mouse until you have to sacrifice the mouse, is only about a week. And that's because these tumors divide, once they get established, they divide every 16 hours. They double in size every 16 hours. And as a consequence, you get a very rapid growth. And I should say, one sees not quite that speed, but you also see very rapid growth in the human BRCA tumors as they emerge. And that's why this is such a lethal disease. Now, so we tried PI3 kinase inhibitors. We tried the pan-PI3 kinase inhibitor BKM120, and it clearly had some effect in this disease, but it was at most stable disease and a little bit of life extension, but no mouse was cured, and within a month, every tumor took off again. If we used the alpha-specific PI3 kinase inhibitor, it hardly did anything at all in this setting. There was a bit of some evidence in some tumors of response, but generally no response. I should say that as these tumors emerged, we were doing full exome sequencing for RNA sequence to determine what are the sporadic events that happen over that 7 months that allow the tumor to emerge. So as we characterize these tumors and also human breast cancer cell lines that were BRCA and p53 mutant to see what happens when you add a PI3 kinase inhibitor, we made a rather interesting observation, which is these are 3 different structurally unrelated PI3 kinase inhibitors that are either pan or alpha specific, and they all 3 activated PARP, an enzyme that causes poly-ADP ribosylation of proteins. And that's what we're visualizing here. And that correlated with an activation of the protein kinase ATR, which is responding to DNA damage. And this is a signature of DNA damage that one commonly uses in the laboratory, the ATR phosphorylation of histone AX at a site where an antibody recognizes that phosphoprotein. And so you can see that adding a PI3 kinase inhibitor causes this DNA damage and that you activate poly-ADP ribosylation, which is a repair mechanism. Now, curiously, I don't have time to explain this, but AKT inhibitors do not do this, or inhibitors of the MAP kinase pathway, MEK, also do not do this. So it's only PI3 kinase inhibitors that do it. Actually, even the beta PI3 kinase inhibitor doesn't do this. And mTOR inhibitors also fail to do this. Now, it was well known that BRCA mutant tumors have excess DNA damage because BRCA is involved in DNA repair, and it's the failure to repair DNA that's correlating with this DNA damage. And so what we're seeing is when you inhibit PI3 kinase, we're exacerbating a basal DNA damage that already exists in these tumors. In other words, they're already having trouble repairing their DNA, and we're causing even more problems for them by inhibiting PI3 kinase. And basically what we were able to show, and I don't have time to explain it, is that PI3 kinase blocks the ability of cells to make ribose at the rate at which you need for making deoxynucleotide triphosphates that are needed to get through S phase and repair the DNA. Now, the concept that you have excessive DNA damage raised the possibility that a PARP inhibitor would actually be useful for treating these kinds of cancers. And the PARP inhibitor, olaparib, had gone into clinical trials even before we did this study, and this data I'm showing you is about 5 years old. But olaparib was already in clinical trials from AstraZeneca and was just approved last year in Europe and is accelerated approval in the United States as well. And this was brought up, the concept that you could push the cell over the edge by impairing its ability to repair DNA by a PARP-dependent mechanism would be a synthetic lethality. So we had some olaparib synthesized and tested in our mouse models, and it worked pretty well, in fact, better than the PI3 kinase inhibitor, certainly better than BYL719, no cures. We could extend life by a month or so, but every one of these tumors ultimately took off again, and we didn't shrink any of the tumors. The tumors are still there, and they eventually became resistant. We're now sequencing all those resistant tumors to figure out what changed to make them resistant to either PI3 kinase inhibitors or to olaparib, and so we're beginning to understand how that works. But here's a remarkable thing. When we put these 2 drugs together, which, as I showed you earlier, causes a massive DNA damage, we kill every single tumor. They completely disappear without exception. And we can take the drugs away after 3 months, and the tumor never comes back. The mice die of old age. So this is really a very remarkable result. which we have never seen. I've worked on mouse models for cancer for many years. We have never, ever cured a mouse before of an endogenous tumor. Now, we don't cure every mouse. Occasionally, we see a tumor come back. In fact, about 20 to 25% of them eventually would develop resistance, but the majority of them are actually cured. We did a bit of cheating here because we actually transfer the tumor that emerges into a litter mate of cleared breast fat of the litter mate so that the other 9 breasts in the mouse don't eventually develop a new tumor, since every single cell in every breast has mutated BRCA and p53. But this is a Kaplanmeier curve now, which is really quite remarkable. You virtually never see this in clinical trials in humans, and it stimulated us to try to do a human clinical trial. We took these data, which we had back in 2011, back to the companies that made PI3 kinase inhibitors. No company made both a PARP inhibitor and PI3 kinase inhibitor, and got AstraZeneca and Novartis to agree to put their 2 drugs together. Either drug had yet been approved, and we started the trial in 2012. First patient was enrolled 3 years ago. The data from this was presented at AACR, and I'll just show you a couple of slides on the consequence of the trial. These are patients, both ovarian cancer patients and BRCA patients. Blue is ovarian, red or gold is breast cancer. And all these patients were not BRCA mutated. The ones who responded best typically were BRCA, but there were some non-BRCA patients who responded. These clinical responses were quite remarkable to the oncologist in that these patients were at end stage in their disease and had failed all other therapies. And what's really quite nice about this is there are a lot of arrows here indicating that the patients are still on the drugs. Even though we've now ended the trial, we have to still keep the drug available for the patients because they're still alive. Some now for 3 years, the longest survivor on this trial. So there's a lot of excitement about this, and Novartis has now agreed to take this into a phase 2 trial in both ovarian cancer and triple-negative breast cancer because I've now spent all of my money and cannot support any more clinical trials. I should say that the reason the companies agreed to give me the 2 drugs is because I was paying for the whole trial. They just had to supply the drugs. But we had a handshake agreement that if we got good results, they would pay for the phase 2 trial. So they did, and I want to finish then by acknowledging, I can't acknowledge every single person who was involved in the work I talked about over the last 25 years, but the people who were involved in the clinical trial and the preclinical data are shown here, and particularly my close collaborator Gerberg Wolf at Beth Israel Deaconess, who was involved in both the mouse model and is also enrolling patients in the trials that I told you about. So thank you very much.